And just a last reminder to all the people who've just joined, you're welcome to keep your cameras on, but please make sure that your microphones are muted so that the speakers aren't interrupted. Okay, so welcome everyone to this very first session of the Biodiversity Genomics 2024 conference. Here, our session today focused on ERGA, the European Reference Genome Atlas. We only have just less than an hour together today, so it'd be absolutely impossible for us to tell you absolutely everything that's going on inside ERGA within this short time that we have together today. But we picked a few highlights, if you like, to try to explain, especially to newcomers or first-time people interacting with ERGA, what ERGA is about, some of our activities and some of our outputs. So we have a couple of speakers lined up for today. Firstly, Chiara Bortoluzzi will be presenting Navigating the European Reference Genome Atlas. She will be followed by Astrid on the sampling side for reference genomes through the community efforts. Then we move more on to the kind of data management side of things with a presentation from Tyler on the ERGA genome tracking console. And then Diego will walk us through some of the latest efforts in building this decentralized approach, if you like, for the community reviewing of genome assemblies. And finally, hopefully there'll be enough time at the end for me to briefly introduce you to our concept of ERGA community genomes and leaving possibly 10 minutes at the end for some open Q&A session. So firstly, just an overview of, especially for the newcomers here, on what is ERGA. So the European Reference Genome Atlas is the European node of the Earth BioGenome project, which many of you will have heard of, this moonshot mission, if you like, for biology, aiming to sequence, catalog, and characterize the genomes of all Earth's eukaryotic biodiversity, with this grand vision for establishing a foundation for biology, aiming to preserve and restore biodiversity and in a One Health approach to looking after humanity as well. And this Earth BioGenome project is, of course, a network of networks, regional nodes being, for example, the African Biogenome Project, and then us in Europe, the European Reference Genome Atlas, but also taxonomic-focused projects like the Vertebrate Genomes Project or geographic-focused projects like the Darwin Tree of Life in the British Isles. In ERGA, you will have possibly heard of the different committees that are supporting all of the different aspects of the reference genome generation workflow. And this involves all the major steps along the way, from the sampling side of things through to the sequencing, into the assembly, followed by genome annotation, and with the downstream data analysis. And of course, this all needs to be supported by IT and infrastructure for managing all of these different steps. But we also take the broader view of making sure to also consider things like ethical, legal, and social issues, media, so how we communicate the importance of genomics and reference genomes to non-specialists. The Citizen Science Committee is also engaged with taking that message to the amateur scientist, if you like, who is interested in contributing to biodiversity, but maybe has less of an understanding of genomics. Then through the Training and Knowledge Transfer Committee, of course, that's incredibly important, especially for our early career researchers, to give them the confidence to be able to perform all of these different steps of the reference genome generation workflow. And finally, our newest committee, the Social Justice Committee, making sure that this is done in an equitable way. In terms of our governance structure rather than our operations, one aspect that is very important to ERGA is this representation across the European continent. So no matter the level of maturity of any country's biodiversity genomics efforts currently, we wish them to be part of this process so that we all walk together towards being in a position in a few years' time where being able to generate high quality reference genomes for the entirety of European eukaryotic biodiversity is possible. So that's what I'm going to say in terms of an introduction, and then I can hand over to Chiara for the first main talk of today. Take it away, Chiara. We see your screen. But we don't hear you.
Yes, now I am unmuted. I hope you can hear me, right? That's perfect. Thanks. Yep. Yes, so I will just give you a broad overview of how to navigate ERGA. So in ERGA, we actually have a vision that includes 3 major points. The first one, we want, of course, ERGA to be responsible for generating reference genome, and all the needed resources that the community needs. As already Rob mentioned, ERGA, we want also ERGA to be an umbrella network for all other projects that are involved in producing reference genomes for all European biodiversity. And then also we want ERGA to be a knowledge hub, so a center for knowledge, and also that includes all steps that are necessary for generating reference genome and also their use. So from, let's say, the beginning to the end of the entire process. So the European Reference Genome Atlas has tremendously increased in terms of representation, in terms of participation over the past year since its establishment. We have more than 1,000 Europe-based scientists. We have more than 200 research institutions across 38 countries. And we have a bottom-up approach, which is, I would say, kind of unique to ERGA, and we are very, very proud of this. In terms of community participation, since we started, ERGA has also increased the opportunity for the community to actively participate in all our work and our, of course, broad and very ambitious objective. We had, of course, several calls over the past years to support financially research projects on biodiversity genomics, so on the use of reference genome and their application in terms of protecting or conserving biodiversity in Europe. We had several projects funded so far, and recently we had several case studies that were financed together with BGE. And if you want to have more information about these case studies, you can visit the website of the BGE project, the Biodiversity Genomics Europe project. And if you are interested to know more about their work, you are very welcome to participate at 3 o'clock, because there will be a session specifically dedicated to it. We are very proud of these case studies because they are not just broad in terms of, let's say, coverage, because they really are, covering very different countries in Europe. But we are particularly happy with the fact that we have very different species involved in these case studies, from mammals to plants to insects and so on. So that also, of course, shows how open we are to really, sequencing everything that is based in Europe, independently of whether the species is charismatic or not, of there's, a specific interest from, many people or just a few people. In terms of community, we have also produced over the past year material, standard operating procedures for sampling, metadata collection and shipment. Also, we have produced several guidelines on biobanking. We have produced several workflows for the assembly of the reference genome, but for any kind of computational work, and several also tutorials and training materials. These are all open access material that you can find in several platforms like Workflow Hub, but also Zenodo and other kind of platform, GitHub, and so on. So if you are interested in any of these, please get in touch with us, and we will be very happy to direct you to the place where you can find all this information. And we hope that they are going to be useful for the entire community. And we are, of course, also open to, update them in a way that they represent also the needs of the community. We also have recently launched this year the ERGA Knowledge Hub. And this is a very open platform that has the objective to collect educational resources in biodiversity genomics. And this is a platform that is curated by the European Reference Genome Atlas community. So you are very welcome to contribute to it just by simply going to the website and then click the contribute button. You can contribute with any kind of material, for example, GitHub pages or tutorials or YouTube videos that, of course, are open access and are going to stay available for a long period of time. You can also choose your kind of contribution. You can contribute, for example, through computational methods and pipeline, but you can also contribute with, for example, guidelines on data management, fair data, or how to engage with stakeholders, and so on and so on. So there are different kind of sections you can contribute to. So you're very welcome to submit your contribution anytime. And in terms of outreach and engagement, over the past year, we have really worked also very hard on this. We, of course, have engaged, you know, in many different settings. And we always try to be present whenever there's the opportunity to actually...
κλείνω λίγο γιατί κάτι προσπαθώ να πάρω.
Okay, do some outreach and engagement. So recently, we had this year, for example, the Long Night of Science in Berlin, where we had our team in Berlin present. And then in terms of other kind of outreach activities, there was a Pint of Science here in Switzerland, in Zurich, where I also presented the work of the European Reference Genome Atlas. We also had, of course, contributed and participated in several conferences over the past year. One of the last where we actually had a very, you know, significant participation of ERGA members were the SMB conference in 2023 in Italy, Ferrara. And also this year, the International Botanical Congress. And one thing that I would like to highlight is that if you would like to participate in these conferences and would like to have also some, let's say, tailored material produced for your audience, we are very welcome to help you out. And actually, we do produce this kind of audience-targeted material based on the activity and the outreach activity that we take part in. So you're very welcome to you know, send us some information or some requests for help. You can, of course, follow us on all kind of social media platforms, on Twitter, on Mastodon, LinkedIn, and also YouTube. We have our own YouTube channel with all the different presentations that were made over the past year and everything that is, you know, coming. So everything is freely available to you. You can, of course, read our recommendations for new articles and also press releases on our, of course, website, but also on our social media. And of course, if you are part of ERGA and you would like your work to be promoted, you can definitely get in touch with us and we will be more than happy to help you promote your publication and initiative. We also pay particular attention to promoting training opportunities, events and resources also on our social media. So also in this case, if you would like us to help you out with it, we are very welcome to do so. So you just have to get in touch with us. So just to conclude, I would like to, of course, shout out to all of you and to those of you who are not part of ERGA yet to just get on board. You can simply scan the QR code here on the slide and just register right now. So we are, as I said, an initiative that is fully open and we are very welcome to, you know, welcome anyone, all nationalities, all career stages. Actually, early career scientists are very welcome to join us and also all areas of expertise. So don't be shy, just jump on board and hopefully we can make a difference all together in Europe. Thank you so much. And I will just leave the floor to Astrid for the next presentation.
Thank you very much, Chiara. And now we're switching over the slide sharing to Astrid's presentation for the second one of the ERGA session. There we go. Yep.
You see it in light mode?
Mm-hmm.
Perfect. Yeah, thank you so much for the introduction. Thanks for joining in today. I will talk about sampling within ERGA. More precisely, I will talk about the BGE, the Biodiversity Genomics Europe project and the sampling strategies that we developed there. You can hear more about that project later today, as Chiara has already announced. We here had decided on 3 levels of involvement of the genomics community to deliver samples to contribute to the visions of ERGA, but also establish a sample to sequencing workflow for the ERGA
Sorry,
community.
one sec, Astrid. For me, it's showing a loading screen still. Okay, so for Chiara as well. So
Yeah.
Shall I stop share?
Yeah,
I
try
would-
to stop and reshare it again. It might be maybe
Oh.
sometimes just
Okay.
So now we see your screen,
Yeah.
yes?
yes.
Try again.
Yeah, we see your screen and now it's loading. And it just seems to be loading.
I would say let's just share the screen, and then instead of have presentation mode, just maybe the normal slides and try to make it bigger. I think it makes more sense. Yeah.
Does it work now?
No, it doesn't. No, it's still loading. So I would just say, Astrid, just share the screen and then the slide, and then not slide mode. Slideshow mode, sorry.
I don't know why, but now it went all gray, the entire presentation.
Oh gosh.
Like as soon as I click share, it goes. Do you see something now?
Yes, now we can see it, but then if you
Okay.
click on slideshow and then it doesn't. So I would say just keep it like that. That's
Okay.
fine. At least we can see it.
Okay, so we tried to deliver samples to contribute to the visions of ERGA and establish a sample to sequencing workflow that would work for the ERGA community. The broadest community engagement was over 2 completely open calls to the biodiversity genomics community, where we asked you to nominate species for sequencing. And this resulted in several hundreds of nominations from a wide variety of organisms and countries. And we worked here in exchange with the ERGA SSP committee on these calls and the selection of species. Our second bucket of species came from 3 European hotspots. So here we work with partners of the BGE project of Spain, Slovenia, and Greece that, however, represent larger national networks from their countries and are all located in the Mediterranean basin. And then sometime into the project, we opened more specific calls for large fieldwork campaigns, so-called bioblitzes, that are now providing many samples for genome sequencing as well as barcoding from further European hotspots. And we asked them to be either focused on a specific region or habitat type on a specific organism. So again, these were open calls in a format of subcontracting funding. And we here selected 6 projects that cover a variety of ecosystems and extended the project especially to the southeastern region of Europe, provided help to widening countries. We deliberately here sampled more than BGE can sequence for now and create a future frozen collection for biodiversity genomics. And you can see some images here. So we really cover many different ecosystems and habitats and different species. So with this selection, we had our target regions and species office, if you like. We then investigated the proposed species and prioritized species, and this was especially true for this very open community-based call. And we worked our way through a species list based on scientific, technical, social, and feasibility criteria to choose what goes eventually into sequencing. Once this was done, we informed the sample providers. They went to the field with guidance from us to obtain samples that would deliver high-quality reference genomes. We asked them to take classical vouchers, or at least an image which serves as an e-voucher to contextualize our genomes. Then different methods of dissection took place, of course appropriate for the various organisms that were sampled. We asked providers to keep material deep frozen. On the one hand, as reference material in a biobank, so for future use and to further contextualize the genomes. And then, of course, the tissues or the material that went into genome sequencing. So this was flash frozen, and we asked them to ensure a cold chain until shipping and throughout shipping to the sequencing centers that tackled the genome sequencing. What we aimed within this project and for ERGA more broadly was a standard for the sampling, but also for the metadata it accompanies. So we asked our sample providers while sampling to collect a diverse set of metadata and a comparable set of metadata. So this worked over a spreadsheet. The spreadsheet is then uploaded to a brokering platform called Copo. Here, all entries in the metadata spreadsheet are validated and controlled against the controlled vocabulary. Once this is fine and the sample manager has approved the manifest, the set of metadata, a biosample is created for all material that is submitted. The accompanying images of the species sampled are brokered automatically to bioimages, and this eventually helps to display all data that we collected, all metadata, together with the sequence data in the European Nucleotide Archive, and this is also visible over the ERGA data portal. And where has this brought us yet? So as of today, or probably yesterday, we collected over 1,000 species. They came, and this is of course oversampling, so this is more than we can currently sequence, but is future-proof sampled and can go to sequencing. We sampled in more than 22 countries. Over 170 species have been shipped to the sequencing facilities that work within the BGE project. And you can see here a short overview or summary of what we have targeted. And we had some deliberately chosen target areas because BGE, for example, focuses on studying pollinators. You can see that we nicely sampled a variety of insects. And here you see how those are distributed across the partner countries. And then, of course, you see that we have a lot more samples coming out of these dedicated hotspot areas that we targeted. And this eventually within BGE is aiming to deliver about 275 gigabases of genome span. What has this effort told us? What have we learned? Community engagement presents challenges, so we have to upkeep with sample providers with the community constantly. But we run within a restricted project, which means we had to enforce very strict deadlines, and this is essential to contribute to the success of a project. But we learned of many, many hurdles that people run into while doing fieldwork. We think we successfully engaged a wide network beyond the BGE project. So we really engaged the biodiversity genomics community. We've learned a lot on coordinating sample shipments across Europe. We've learned that this is complex. We hope that BGE can highlight this and show that there's improvement needed, that facilitates our work together. This project delivers clear workflows and SOPs that are hopefully transportable to other projects. And we've learned very quickly that automation is needed for this wealth of metadata that we produce, but this is a workflow that we have established, and we're very happy for every ERGA attached initiative to use that. And with this, I'm very sorry for the screen sharing issues. I hope Tyler will have a better time, so I'll stop my screen.
Thank you very much, Astrid. And while Tyler is sharing his screen, yes, so a lot of the things developed in the Biodiversity Genomics Europe project that Astrid was presenting there, as she said at the end, are exactly translatable, transferable to the wider ERGA community, national projects, taxonomic projects, etc. Go ahead, Tyler. So now we move from sampling into the data tracking.
All right, can everyone see my screen?
Yep.
Perfect. First hurdle done. So my name's Tyler. I work at the CNAG in Barcelona, and I need to figure out how to advance the slide. There we go. So I lead a genome assembly and annotation team, but within ERGA, I'm also helping to coordinate a lot of the other assembly teams so that we can deliver, a whole bunch of gigabases of genome span for the BGE project. And so basically this requires handling a lot of data, all the sampling data that Astrid was just talking about, through the DNA sequencing, the assembly, annotation stages. And this has to be done in a context of a continent across Europe, and we have sequencing centers to the north, to the south, in the middle. And the work is highly distributed, and the expertise is highly distributed, and the samples are coming from all over. And so the question is, how do we coordinate this specifically? Okay. And so do we use spreadsheets? Astrid already referred to one spreadsheet. That would be the ERGA manifest, right? This is very comfortable for biologists. We're all very familiar, especially laboratory bioinformaticists. We work with spreadsheets all the time. This is good for collecting information. It's a comfortable way to input data. It's a nice way to collaborate, and it's a nice way to kind of visualize what you have. But over time, it starts getting out of control. Okay, so these are various spreadsheets that have been passed around during the course of the project, and they're very useful in smaller groups. It helps coordinate, you know, what species we're going to prioritize, who's going to do what. But if you look at the entirety of them, the columns switch, the color coding switches, there's cross-throughs, there's text in red, lots of notes. This is not a way to visualize all the data at once, right? So the idea that we had, my idea, and it was backed by many other people, is to take these data and put them into a single DBMS, or a database that can be accessed by all the parties involved across all the institutions, one place where we have all the data represented. This is not eliminating all the spreadsheets. There's still some communication behind the scenes, and it's very useful temporarily to have spreadsheets, but we are collecting it all in one place. It's called the ERGA Genome Tracking Console, not Tyler's tracking tool. Sorry, the GTC. And so what it does is, behind the scenes, it can connect to COPO. It can import all of the sample data that has been submitted and approved by the approval committee, right? It can connect to GOAT, so we can send them our target list. And in return, we can get from GOAT a lot of the genome characteristics that GOAT is very good at collecting. And so we can pull in the estimated genome spans and karyotypes, these sorts of data, so that we have it alongside the species that we're working on. It can connect to the ENA and figure out what's been submitted and what is left to be submitted. This is more important for us to figure out what needs to be submitted, okay, what's lacking. The public data portal that's being developed for these projects is very good at showing the public everything that is finished, but we internally need to know what needs to be done, where is it, how can we push it forward. And any of the other actors involved, the sequencing centers, can also interact with it via the REST API and upload information on runs. The assembly groups can upload information on assemblies, and this can all be automated. But some people still like to enter things manually, so there's an admin interface for entering the information manually. There are forms that we've developed to upload species lists, and with the species that we're targeting, also the information on who is supposed to collect it and by when. There are interfaces for managing the genome team, so the sequencing centers, who's going to sequence it, who's going to assemble. And so this is the main status page right here. Okay, and it shows the state of every genome that is going through the pipeline. And so the pipeline is starting with the samples and what's been accepted in COPO, how much long-read data has been produced, any short-read data, high-C, RNA-seq, whether assemblies are under review or have already been submitted. And this is view only or read only. And but you can filter. It's highly flexible. Every view here has a filter. So it's a powerful way to query the data and drill down to what you want without looking at all the records in a spreadsheet. If you are a sample manager, let's say, you might start with uploading the species list, being able to edit the information in the console. Would show up like this. The species here. Behind the scenes, there's another table that gets sent to GOAT with the sequencing status and the target list status. And the sequencing status is updated automatically in a lot of cases. So if something is already being generated, data is generated, it'll change status here. Okay. And a couple other views, drilling down to the specimens and the samples. So all of this is pulled from COPO. If you are a manager, you can set all the teams. So you can say who is doing, let's say you already know who the sample coordinator is. The sample coordinator may enter the information on who did the collection, who did the taxonomy identification, where the vouchering is being done. And someone from a different work package might say, okay, they agree behind the scenes that SciLife is going to sequence this first genome here with the long name. Okay, and CNAG will sequence this cockeyed squid in row number 2. And we can collect information on all the runs, including the yield and N50 and quality. This can be summarized, so we know all the data that's been produced. We have the self-reported data, and we have the data that is pulled from the ENA, so we know exactly what's been submitted. And the assemblies as well. These can be self-reported or pulled from the ENA. These are all in the ENA, the ones shown here. And after the fact, if you want to figure out who did what, when it's time to produce a genome note and publish, there is a list of authors with their roles, and some authors have more than one role, so they're listed more than once, right? This is a nice way to collect the information, and we'll see if we can use it more in the future. And it's hosted here. There's a production instance that everyone is using for development. We have another one, and the code exists in GitHub and will be forked pretty soon to the ERGA GitHub. Okay. And the code is welcome to be used by any EBP initiative, anything under ERGA, for sure. We'll have an instance for the Catalan Biogenome project. And it's very flexible. If you have sub-projects, let's say the pilot or BGE1, BGE2, that's possible within the system as well. So I'm going to stop and hand it over to Diego.
Thanks a lot, Tyler. Yeah, there we go. Thanks. So as I said at the beginning, we're not going to be able to cover every single step of what we're doing inside ERGA. So now we're going to jump over the assembly of the genome assembly part and go immediately to the reviewing of the assembly, which Diego will tell us about now.
Can you see my screen?
Yep, all looks good. Go ahead.
Beauty. Okay, let me just close this thing. Yeah. Thank you very much. Hi, I'm Diego, the ERGA Sequencing and Assembly Committee Coordinator, and I will talk you about a nice thing we produce in hopefully 8 minutes. This is about a decentralized method for community reviewing Earth BioGenomes. So, yeah. So the reality that we face in our project, and I think this is extensive to many others, is that the genome production is distributed across different sequencing centers and institutes and labs in different countries. All these colleagues are working on many different genera, using a variety of combinations of sequencing technologies, producing different throughputs and qualities, and using their own selection of tools and pipelines to process and evaluate this data. So there is a lot of variability about how to get there, but at least we know where we want to go. We want to produce high-quality reference genomes. And importantly, we know what a high-quality genome should look like. Okay, we define this using the UP assembly standards. The standards are flexible, but this is the B5. Now we have the V6, so I will update this soon. But yeah, I was telling you that the standards are flexible and they adapt to the sample source and taking into consideration the organism type don't see this as an impossible thing to achieve. So we need something to help us to quickly assess the quality of genome assembly in a standardized way, but something that all the different persons involved would like to use. So the solution should not be imposed. The solution should be agreed. And ideally, a solution should come from the community. So at the ERGA, we place the community at the center. A lot of what we do is related to community building, providing places to exchange ideas, presenting new methods and updates, let people show what they have, drop questions, and so on. Examples of this are initiatives like the Summer Assembly Clinic or the workflows that the community is contributing in Workflow Hub. This is something that Chiara mentioned. And we want to consolidate and expand this network. We want the community to produce useful things for all. So we asked the community which QC outputs are essential to evaluate quality of a genome assembly, keeping it simple based on widely used tools. And with all this information, we produce the ERGA assembly report, the EAR. It's a standardized PDF showing relevant information to confirm that the required quality is met. So this is what the EAR looks like. The PDF is a simple report, always in order, contains information from different sources, not from the curator, retrieves data from Gov, from NCBI, presenting agreed metrics and analysis, a link to a high-sync contact map. And this is the other part, showing Koenig plots, contamination screening, the tools that person used. And we have a wiki where we explain everything about this PDF and how to create one. And please check it. And we provide a script to do it on your computer or server, or to add it to your existing pipelines. And also with the fantastic collaboration of Saim from the Freiburg team, we ported this into Galaxy, so you can extremely easy do this on Galaxy. So you need to run all the QC programs on your assembly and using this to generate the report based on all those outputs. Okay, so now we have these standardized reports to help us to decide if a genome is high quality. So the next thing we need is a reviewing system. We need to connect this report produced by a researcher or a sequencing team that did the assembly with an external reviewer that will look at the report and will confirm that it is good. So what we did, again, trying to keep it simple but robust and with the feedback from the community, we came up with a system that happens inside GitHub, so everything is open, transparent and traceable. So to start the process, a researcher opens a pull request to the EAR GitHub repository with the PDF of the assembly review, to the assembly 2 review, and that pull request becomes an isolated conversation for that assembly where everything will happen. If all is good, the report will be approved and you are good to go to upload your genome to ENA or another repository. And if not, together, we will try to fix the issue and until everything is good and you can upload your genome. So all this reviewing process will involve the following participants in this conversation: a researcher, the person that is behind the process, behind the genome assembly and submits the report, a bot that will handle many tasks during the process, a supervisor that will jump in if needed, providing moderation and overseeing stuff and other functions. And of course, a reviewer that is going to be the person that will check the report and ask questions or clarifications to the researcher if necessary. And again, all these people are from the community, doing things for the community. The bot is not a person, but a person was instrumental in its implementation. This was Arash, again, from the Galaxy team in Freiburg, led by Björn. So the process looks like this. the researcher opens a pull request, adding the PDF to the assembly review, and the conversation starts. The bot is going to run some checks, add a tag, communicate with the researcher, and call the supervisor. And here you can see a real example. Like this is from Benjamin from Genoscope. He sent an ear for this species. I think it was a seagrass. And yeah, it goes as I told you. Conversation goes in an open and traceable way. And then after the supervisor accepts, the bot is going to call a reviewer from a different institution, from a pool of reviewers from the community. This is the list that you can see here. To keep things fair and balanced, the selection is based on points that change with the amount of reviews that a person did, how many PDF that person institution sent for another to review, also based on the last review done. So if an institution is asking frequently for someone to review assemblies, it is more likely that a person from that institute will be called when someone needs an assembly, okay? Then when the reviewer accepts, it's assigned to that report, and we'll check that year and look at the contact map, and we'll drop a message according to what is found. Here, Sarah from Sanger is pointing out some things to improve. And as you can see here, the process is very dynamic. It goes back and forth, the chat. You can attach images to explain your point. And very importantly, we are seeing that this is very educational and transferring knowledge. It's doing a lot for balancing the knowledge among partners. So finally, the reviewer approves the report, and the supervisor adds the PDF to the stable repository of reports. And the researcher is good to go to upload the genome to ENA, okay? So in addition to the assembly that is now available on ENA, you will have the year as a PDF in the stable repository of reports with curation information, and also all the conversation that happens during the review, providing context. Everything traceable, everything open. And to close this presentation, I just want to say that I just show an example of a community-driven solution, and I wanted to highlight this because we are facing challenges that I think cannot be solved exclusively by individuals. So I wanted to vindicate the collective efforts, but without ceasing to recognize people individually and their contributions to the community progress as a whole. So we would like to see you around in the ERGA community. I think together we can do cool stuff. And yeah, we can find each other in different spaces. Please save these links. And that's all for me. Thank you very much.
Thank you so much, Diego. That's really, really cool stuff. Okay, so I've got to try to keep moving for the last presentation, which is from me in this session, kind of connecting some of the dots that you've been hearing about up until this point, here focused on what we call the ERGA community genomes. So I think you will have seen in the presentations that came before that the genome generation workflow contains many, many different steps where you can go wrong, where you can fall down if you don't have a helping hand along the way. And really, this is the motivation of the work behind building the ERGA community, to be able to immediately find someone who has experienced these challenges before, who has maybe found a solution and can help you overcome your issues, all the way from the very beginning of the stages, through the sampling, through the extractions, through the assembly. You just heard about the reviewing process to ensure quality, all the way through to the data deposition in the public repositories. And really, you know, how this happens and where this happens, and you heard there briefly from Diego about the work of the sequencing and assembly committee as one of these pillars, is the committees in ERGA, where these are the places, these are the forums where we are trying to develop these community standards that are agreed upon by participants so that they are realistic and achievable. But also practical things like best practices documents, practical guidelines. You also saw some examples of bioinformatics analysis workflows where and Diego even said there that they've been ported onto Galaxy so that they are widely accessible and that everyone can be using the same kinds of workflows, the same kinds of standards, so that we're talking the same kind of language, so that we know what we're talking about when we are addressing things like assembly quality. So metadata you heard from Astrid's presentation earlier, information sharing about what works and what doesn't work. And this is really the fundamental work that is carried out by the ERGA committees. For example, here as a kind of starting point for the community genomes, we put together a list of questions that a researcher might ask themselves at each different step along the process. Again, with some answers to those questions, but usually directing to the relevant committee where that question might be posed and hopefully someone can answer. So for example, just under point number 2 here, under sample acquisition strategy, have you thought about your permits and ABS? Have you thought about your permissions and your material transfer agreements? How are you going to do your taxonomic validation? Do you know what is specimen vouchering and where you might be able to do that? Have you thought about where you might send your leftover samples for biobanking? Do you know the best conditions for shipping? So for example, after you've done your collections, to send it to the sequencing center. You can ask these questions, find the answers so that you don't end up shipping it incorrectly and your sample arrives at the sequencing center in a pool of melted ice. So that's just one example of the kinds of things that we're trying to implement to help community build their own reference genomes. Part of this is, of course, also coordinating efforts across different projects, across different research groups, in our case, at the European level. And so this view here that you can see on this screen is the portal, ERGA Biodiversity portal, which is tracking biosamples created at the European Nucleotide Archive, which Tyler mentioned earlier. So the projects that are coming up, the samples that are arriving, and the data that are arriving, and eventually the assemblies that are arriving, so that the entire community can see at what stage the generation of a reference genome is at for different species being produced by a whole group of different projects underneath the umbrella project of the European Reference Genome Atlas. And for example, another important reference resource, if you like, for tracking and coordinating efforts across Europe is the Genomes on a Tree resource that Tyler also mentioned earlier. And here just an example of specifically the Biodiversity Genomics Europe sub-project of ERGA, showing more or less the progress in terms of what has been collected, what is undergoing sequencing, and what is moving into the assembly process. And so how, is the best way to begin to engage with the community coordination efforts? And really this boils down to two main points, is how to communicate which species you or your research group is working on at the moment. And then at a second stage, how to get your genome ERGA approved, which is something that Diego was addressing there with this assembly review process. So first of all, communicating which species you're working on. This is mainly through the Genomes on a Tree spreadsheets. And at the kind of ERGA coordination, we're trying to centralize this to help people to achieve this more easily. So either through the spreadsheets that are as part of the BGE project or the pilot project, so ERGA core projects, or other European projects that some of them were mentioned today, the Catalan BioGenome project and others that are related and connected to ERGA. And what's currently under development now is building country-specific sheets for Genomes on a Tree, so that each country can really showcase to the world the different species they are working on, or the research groups that which taxonomic groups they're focused on. And in parallel to that, building what we call the ERGA community Genomes on a Tree sheet, while we're still getting established to have individual country GOAT sheets. In terms of the second stage, which is maybe you've already produced your reference genome and now you want to get it ERGA approved, here it's really two simple validations that we are implementing at the moment, which is the first one, the metadata validation check. So this is basically the samples and sample processing committee checking that your metadata that you've collected meet the minimum required standards to be considered an ERGA approved genome. and the assembly validation. So those are the EBP standards that Diego was just presenting there. So the sequencing and assembly committee being in a position to check that your assembly meet the required standards. Then we can move or connect your bio project on the European Nucleotide Archive underneath the ERGA umbrella and giving it that ERGA seal of quality or ERGA approval, if you like. And finally, how to do this. So when you get stuck, when you have questions, really the main take-home message of this session is that there's absolutely no need to struggle all alone. We have within ERGA our Keybase community chat platform, and we also have for all of the different committees group mailing lists. So those are kind of your two main avenues of communication with experts who might be able to help you answer your questions to overcome your problems. So this is also to reiterate that you don't have to be a regular committee participant. So you don't have to be turning up at every single monthly committee meeting in order to ask for help. So through either the Keybase community chat platform or through joining the mailing groups of each of the different committees, you can post your question on Keybase in the committee's relevant channel, or you can email the committee steering group, which each committee has its own email address, which you can find clearly on the website. And indeed, you could even perhaps request a slot in an upcoming committee meeting where you might present some of your data and say, this is where I've got. I don't understand what these graphs are telling me. Could you help me understand if I need to go and do more sequencing or if there was something wrong with my sample? And there's a group of experts, and amongst this group, hopefully one or two of them may have seen something like this before and can give you advice on how best to proceed. So if you haven't signed up to join ERGA already, then hopefully you are motivated to do so. And the channels there are really easy in the Keybase community chat platform and the committee group mailing list if you want to become more engaged. And you can also just browse on the website for the time being to find out the kinds of events, the calendar of events indeed, for when each of these committee meetings are happening. So Chiara started off by saying we welcome interaction, we welcome participation, and I guess I can kind of close the circle now with the reiteration of that message. And thank you all very much for joining us this afternoon. I believe we might have time for one or two very short questions from the audience because we're a little bit over our time. So if you have a burning question, then please feel free to raise your hand and we'll see who can go first. Okay, I see lots of hands clapping, but I don't see a raised hand. Okay, so in that case, we finished perfectly on time. I hope that gave you some insights into what we're about and who we are. Again, there's a lot more things going on behind the scenes, so please do join us. We have our monthly plenaries where we have invited speakers from all sorts of different positions along this genome generation workflow, from the very beginning to the very, very end, and indeed applications. The committees also have some very interesting presentations in their monthly meetings, usually on much more focused topics. So there's a lot more to learn, and please feel free to join us. Thank you very much, everyone, and enjoy the rest of the Biodiversity Genomics 2024 conference. Take care.
Thanks, Rob.
Bye, bye-bye.